my name is Gea. I'm a PhD student in the Bio-Inspired Photonics group.
And I'm Tom, also a PhD student in the Bio-Inspired Photonics group. In our lab, we study how living organisms use nanoscale structures to create color, and inspired by what we see in nature, we use sustainable materials from natural sources to also create colorful structures.
Let's go and have a look in the optics lab. In this lab, we characterize the visual appearance of our samples using optical microscopy and spectroscopy.
So on the microscope here, we have a film made of cellulose nanocrystals, which are rod-shaped nanoparticles that we extract from natural cellulose. If we could zoom in and see the structure of the film, it would look something like this, with these layers of rods all stacked together in a left-handed configuration. And as a consequence, this structure only reflects what we call left circular polarized light. And we can see that on the microscope here. When you look at the camera shot here, you can see that the film appears a lovely blue color because we're filtering for the left-handed light. But if I change the settings so that we filter for the right-handed light, you see that the film in the right-handed channel only reflects a dull brown color. And this is a response that we see in natural structures, and it's something that we can recreate in the lab using our cellulose nanocrystals.
As you can see here, structural color is often iridescent. This means that the color you see depends on the angle at which you're looking at it. This is why we characterize our samples using an angle-dependent spectrometer. Structural color occurs across the entire kingdom of life. It occurs in birds, such as the peacock, which has the nice colored tail feathers, and it also occurs in beetles, such as these green and red beetles. It also occurs in land plants, which have these structurally colored fruits you see here, and also in algae. In addition, structural color even appears in microorganisms, such as these iridescent bacteria. These green beetles are actually the biological inspiration for the material we looked at previously under the microscope. And if we look at them through these 3D glasses, which has two different types of polarization filters, through one side of the glasses we can see the color, but through the other one we cannot.
Inspired by what we see in the natural structures, we can use sustainable materials to make color in a range of forms, from large-scale films to photonic pigments and glitter. This color is biocompatible, biodegradable, and even edible. We're also looking at making advanced photonic structures such as holograms. That's everything we wanted to show you in the optics lab. Now let's go to the wet lab, where we make these materials and grow our biological samples. In the wet lab, we have everything we need to work with colloids and polymers. For instance, the cellulose nanocrystals that we use to produce the colorful films are extracted from cotton by acid hydrolysis. After this, we purify the nanocrystals using dialysis, as you can see here. The result of this process is a cloudy suspension, which we use to make the colorful films.
In addition to structurally colored algae, we also look at other species of algae to study their light management in bioreactors to optimize their growth.
Thanks for watching this lab tour from the Vignolini group. If you'd like to know more, check out our website.
We hope to see you in our lab soon.